Hi, everyone. My name is Arseny Finkelstein. Thank you for the opportunity to speak here. I'm broadly interested in how neural interactions give rise to cognitive behavior. And today I'll talk about how cortical interactions give rise to goal-directed behavior and transitions across brain states. So, a major goal in neuroscience is understanding the link between neural coding and the underlying connectivity. And there was significant progress in this in sensory areas such as the visual cortex, understanding the contribution of recurrent connectivity and inputs from across different brain areas. But as you go to high order brain areas such as the hippocampus, this becomes more and more challenging problem. And this problem is also very challenging in areas in the frontal cortex, such as the motor cortex, because partially the activity is internally generated. And the other challenge is that contrary to, let's say, visual cortex or the hippocampus, where you can observe the neural representations just by presenting a particular sensory stimuli or letting the animal doing natural activities such as the navigation. In the motor domain, you typically need some form of a task to let the animal perform, and through this task, learning about the underlying neural code. And this requires essentially some form of training, and as a result of training, likely the underlying neural connectivity and the representation it would evolve. So we don't know much about the sort of native coding scheme in the frontal areas in naturalistic behavior to a large extent. And we try to make a step towards understanding the native coding scheme in the frontal cortex, particularly in the motor cortex, inspired by the primate literature to develop tasks in rodents that would have rich motor repertoire but would not require any training. So in this task, the animal is presented with a target that it would lick for a water reward, and the target can be presented in different locations around the mouse face, and the mouse would simply need to collect the reward. And the advantage of this task is that it has a relatively rich motor repertoire, and it doesn't require any training, so mice can perform it from day one. And in this task, in the motor cortex of untrained mice, we see a very robust encoding of target location. So this is one example neuron would have a location tuning, and this neuron prefers to be active in the upper central part of this grid. And these are examples of 100 simultaneously imaged neurons in the motor cortex. Each neuron would have a different tuning pattern, so they, to some approximation, look like place cells for this target location in front of the mouse face. And these neurons are also modulated not only by the target location, but also by the reward that the animal would receive in this different location. So they encode the action and the outcome of this action. And by combining calcium imaging and targeted photooptogenetics, we were mapping the underlying connectivity and relating it to this type of neural code in the motor cortex. You can get very detailed connectivity maps in the behaving animal using this system. And we established a relationship between the connectivity and the coding. For example, like-to-like connectivity between similarly tuned neurons. This is a work that would appear next week. And today's talk would basically build upon this behavior and explore the multi-regional interactions underlying these naturalistic forms of behaviors. I would speak less about the local recurrent connectivity and would sort of zoom out to the large scale cortical interactions. So when we just observe the mouse in this behavior, you can see that before the animal licks, there is an extensive movement. For example, the animal moves its whiskers. So we might assume that there are some interactions between the somatosensory cortex and the motor cortex, for which I showed you the tuning. And in fact, when we look at the neural activity, and this is imaging of about, and I think you don't really see it. You might not see it well, but what you see here, I think it's okay now. Thank you. Yeah, thanks. So what you see here is imaging, calcium imaging, using a two-photon mesoscale imaging, where every pixel here is a single cell. So here we are imaging about 30,000 simultaneously recorded neurons. And each of these neurons, if you, for example, look at this secondary motor cortex, would have this precise tuning to location and reward. But if we just look at this collective activity of these 30,000 neurons, we see very diverse and complex patterns. So we wanted to understand, without actually trying to focus on the individual tuning properties of the neurons, the interactions between the different cortical areas during this task. So this is a work that is spearheaded by Lee Zussman, a postdoc in my lab, in collaboration with Friedrich, Henning, Carl, and Jonathan. So in fact, this work is supported by our collaborative grant with Jonathan Al-Khadef through the CRCNS, and Jonathan is here. So first, we wanted to understand the dimensionality of the representations in this task, and we heard several talks about dimensionality, and there is a very influential assumption that activity during the task, during behavior, is low dimensional. This is supported by many studies, and these low-dimensional representations are also very appealing because they provide an intuitive link between neural population activity and behavior. And typically, this assumption about the low-dimensional nature of the task comes from trial-averaged neural activity. So here again, instead of trial-averaging neural activity and looking at tuning, we first ask, in this task, across the about 10 different brain regions that we image simultaneously, spanning motor areas, somatosensory areas, and high-order visual areas, what would be the dimensionality of neural activity? And what Lee found was that the neural activity was very high dimensional. So we have, for 30,000 neurons, about 1,000 reliable dimensions of activity that are needed to explain about 90% of the variance. So despite the fact that the task is pretty simple, there are about 16 targets the animal has to lick. Each one of them, we have very high dimensional neural activity during the test. So the behavior is conducted during darkness, and we thought that maybe the dimensionality of, or as a measure of complexity of the activity in each area, that this dimensionality would be different in areas that are actively sort of engaged in the task, for example, the motor cortex, but would be different from visual areas that presumably the animal does not use during this task in darkness. So for example, we thought that maybe the activity in the motor cortex would be more low dimensional during the task performance. But what we found that, in fact, the dimensionality was high across all regions sampled, was rather similar across all of them, and the dimensionality scaled with the number of neurons. And so the more neurons we took into consideration, the higher was the dimensionality, and this relationship was very similar across all areas, despite the presumed role in task engagement. When we look, for example, in two different areas, in the barrel cortex and in the secondary motor cortex during this task, we see that their mean activity is different. So the barrel cortex is active before the reward, the motor cortex is active on average during the reward. But when we look at the dimensionality in this time slices during this particular trial, we see that the dimensionality falls down, essentially collapses to a large extent around the reward time. So it's still, instead of 1,000 dimensions, we are getting about 500 dimensions. So it's not an absolute collapse, but there is a significant reduction. And this is something that is observed across all area samples. So despite variability in the mean firing rate during the task, all of the areas exhibit this quenching of the dimensionality around reward time. And this is related to the phenomena described by Mark Churchland about the stimulus onset that quenches neural variability across different brain regions and behaviors. And if we just look at the trajectories that these different regions exhibit, when one of the axes is the mean activity and the other is the dimensionality during these trials, we see here an example of two mice for barrel cortex and secondary motor cortex. We see very characteristic dynamics that these brain areas have, essentially a footprint in this dimensionality and the mean activity space. What we found is that this collapse of dimensionality is reflected essentially by global synchrony across brain areas around reward time. Another finding that we saw was that during spontaneous activity, that basically we imaged the same neurons before the task, the dimensionality was even higher than during the task. And we asked ourselves whether this reduction in the dimensionality during the task is attributed to increased synchrony or coordination within each area, or in contrast, that each area keeps its dimensionality high and complex, but that the coordination between areas is increased during the task. First, we measured the dimensionalities across these two brain states for different areas and found no difference. So basically, the local complexity across each area is similar during task and during spontaneous activity. The pairwise correlation, just the average pairwise correlation between cells, is also very similar. So this is a very robust result. We have a huge number of neuronal pairs. And when we looked at the distribution of pairwise correlation as a function of anatomical distance, we also saw, despite a big heterogeneity across brain regions, so some regions have more prolonged distance-dependent profiles, and in others, the correlations drop faster with distance. Across states, this distance-dependent profiles are very similar. So brain states have similar, on average, pairwise correlations and similar distance-dependent distribution of these correlations. But when we actually look at the correlation structure across the two states, they are rather different. So the average similarity is about 0.4. So different states, despite this invariance to dimensionality, dropping correlations and average pairwise correlations, have very different structure. We asked whether this overlap in the correlation structure is low, high dimensional across the states, and we found that actually these two brain states have a very low dimensional overlap. So basically just about two dimensions that carry an overlap between the states. And these dimensions are mainly influenced by animals' movement. So the regression of the movement against these two dimensions that are shared between spontaneous and movement and task-related activity is carried by the movement signal. So we can say that this reduction in brain complexity during task is not attributed to local shrinkage of within area complexity. And we ask more formally whether the interactions across brain areas contribute to that. So for that, we measured the interactions across the different regions. We adopted the communication subspace framework that was developed by Semedo and from Adam Cohen's lab, who is here at the meeting. So we measured essentially the dimensions that are shared across source and targets across these different regions. We found that the communication subspace explains about 20% of the variance across regions. That is high dimensional. The dimensionality is increasing with n number of neurons, and the communication subspace is stronger during tasks compared to spontaneous, and we also see a distance-dependent profile of the communication subspace. So close by region communicate more, and this communication is increased during the task. We also found that essentially through this synchrony across brain areas, the overall complexity of the global dimensionality is shrink. And again, whether the local dimensionality and complexity remains the same across the tasks. So I would just skip that and summarize that we found out that the cortical activity is high dimensional during tasks, scales with n, and quenches around reward time. The global dimensionality is even higher during spontaneous activity, but local within area dimensionality remains invariant across states, and this is ruling out reduced local complexity as a cause. The internal communication is high dimensional. I didn't show you, but it's actually multiplex and increases during tasks consistent with enhanced global synchrony. And across states, despite these invariances, the cortical dynamics share a low dimensional backbone, suggesting a stable scaffold enriched by space-specific activity patterns. And again, this is work that was led by Lee in close collaboration with several people, particularly with Yonatan Al-Khadef, and thank you.
Time for a couple questions. I'm wondering, does the communication subspace explain all of the reduction of the dimensionality, or do you have some dark matter that is not explained?
So it's an interesting question. We can say that pairwise, the communication subspace explains about 20% of the variance on average. But if you take several regions, that would explain about 0.5. And this might be sufficient to explain this reduction. We should relate it more closely, but yeah.
Is the scaling of the dimensionality and consistent across mice, and is there any model that gives rise to this?
So the results were robust across mice and actually across regions and each mice. And the scaling factor is about 0.25. So for every 4 neurons, you basically add a dimension. In terms of the model, we don't have a model at this point to explain this, but we think that essentially, if you take into account the structure of pairwise correlations, the local interactions that would contribute to the scaling. And in fact, as you go to larger areas from local to global complexity, the scaling factor changes. And that's exactly where we think that this cross-areal synchrony kicks in.
Okay. Thank you very much.
Thank you.